YouTube is catching up with us as well. Lovely. So this seems to be working. Everything is going well. Hello and welcome everyone to our CNS talk today. The first one after the summer break. And it's nice to see so many of you today. Today we have a fantastic speaker. She has a double background, MD, PhD, which she managed to acquire in the space of 10 years of MD, PhD at Tübingen, specialization in neurology, plus the habilitation, already on learning processes in the cerebellum at the University of Essen, where she's now a professor for experimental neurology. And just to give you a scope of the impact of our speaker today, she has 16,000 citations over the last 20 years, and her research output only for this year didn't even fit on one computer page. So we have many topics to look forward to. And I'm very excited to welcome our speaker, Professor Dagmar Timmann. I hand over to you.
Yeah, thanks a lot, Stephanie. Thanks a lot for the invitation. I will change to my presentation here. Can you see it all right? Including my mouse? Okay, so I put together 25 or 26 slides, so I hope that I stay in time. And what I do is I will present data of mainly motor learning paradigms, but at the end, also one emotional learning paradigm we did over the years, mainly in patients with cerebellar disease, but I will put in some functional MRI studies as well. I like to start my talks with a case of a patient who has been born without a cerebellum. So worldwide, I think there are 10 or 20 cases around now, with people getting MRI scans frequently and easily. Numbers are slightly increasing. So you see an MRI scan here of this patient, and in the posterior fossa, cerebellum is almost missing, except for a very small piece. This patient, she has motor dysfunction, but surprisingly little. So when you look at her arm movements, there is some dysdiadochokinesia, but not that much. And here you see her, this is how she stands. She has a balance deficit, but surprisingly little, given that she has no cerebellum at all. She needed longer to learn to walk as a healthy child. And what I will do first, I will show data of eye-blink conditioning, and I will show data of eye-blink conditioning in this patient born without a cerebellum. Eye-blink conditioning is a simple associative motor learning paradigm, which has been used for many, many years in the field to understand different aspects of the cerebellum's contribution to motor learning. And I guess most of you will be aware of it. In eye-blink conditioning, frequently an air path is used as the unconditioned stimulus blown to the eye or next to the eye. And the conditioned stimulus is frequently a tone presented to the ears. And usually simple delay paradigms are done with a CS presented for example, in humans around 500 milliseconds and an overlapping for the last 100 milliseconds in US. And here are 100 trials, paired trials, CS-US trials, with EMG recordings from the eye muscles. And at the beginning, there was only EMG activity related to the US, which is the simple eye blink, brainstem evoked. And then after 20 or 30 trials, there are conditioned responses. So EMG responses after the CS, prior the US. And in this patient born without a cerebellum, she doesn't develop any conditioned responses. So she has unconditioned responses. So the blink reflex itself, brainstem circuit is fine, but she doesn't learn that the CS predicts the US, and she never closes her eye prior to the US. So this was the case, and same thing is seen when you look at larger patient populations. It's a very sensitive paradigm, so it doesn't matter what kind of a disease you have. So in this study, we had patients with degenerative disorders, SCAR6, and idiopathic cerebellar ataxia with more diffuse cerebellar degeneration. And these are these dark dots, and these are 10 blocks with 10 paired CS-US trials each, and simply the number of conditioned responses is shown here, CR incidence, and there are almost none in these patients with cerebellar degeneration. Then we tested patients with stroke. So this here means superior cerebellar artery stroke, and they perform not much different from the patients with cerebellar degeneration. And these open dots are patients with strokes in the territory of the posterior intracerebellar artery, and they show some learning. Here, it's just all the patients grouped together. So these dark circles are all of these patients grouped together compared to a control group. And in the control group, there is faster learning and more learning. This comparison of patients with pica stroke and SCA stroke shows that not all areas are involved in enabling conditioning. Same thing as in rodent or other models. And just one example, this is pinning this down, which areas are involved in acquisition of conditioned enabling in humans. This is a study we performed in young participants around the age of 20 or 25. And in the patient group, the cerebellar group, they have surgery for benign cerebellar tumors, so they have surgical lesions in the cerebellum. And when you look at all of the group, again, cerebellar group performs worse than the control group. In this paradigm, we had an acquisition phase followed by an extinction phase, and we had a reacquisition phase. Then we subdivided patients and controls in those who were able to learn the enabling conditioning paradigm and those who were not. So these dark dots are the controls, and some of the patients, the open circles, perform as good as the patients, and then there are very few controls who don't learn, and then there are many patients who don't learn here. And then when you look at the lesion sites, comparing the patients who learn and who do not learn, there are differences. So this is simple lesion overlap. So in the patients who do not learn, surgical lesions, maximum overlap is in the more intermediate parts of the cerebellum, in lobule 6 extending to crus 1. And patients who are able to learn, they have lesions more in posterolateral areas of the hemispheres. And these intermediate parts of the cerebellum, lobule 6, with extension into crus 1, this matches very well with what is known of the rodent data in mice or in rabbits. So there really the hotspot for enabling conditioning, one of the main hotspot is in lobule 6. This is all about acquisition. What about retention or the memory in the cerebellum? When you do studies in cerebellar patients, it's very hard to look at retention because they don't learn in the first place. So we did one paradigm. We looked at a form of associative learning, which is done naturally in the very first months of life. And the idea was to use this paradigm to test this in patients who have acquired their cerebellar disease in adulthood. So the idea was we use this paradigm, which I will explain shortly, where we have a conditioned response which has been acquired in a natural way in the early months of life, where we think that these patients had a normal cerebellum, so they were able to acquire it, and then we looked at it 40 years later when they had developed a cerebellar disease in order to look if the cerebellum has something to do with the storage or memory of this learned association. And the paradigm is called visual threat visual threat paradigm. And the idea is the following. So this is a healthy baby, 1 month old, and Michael Küper worked as a postdoc in my lab. And he and his daughter, she is now even older than 77 years old. So they said, yes, I can use this video. So Imke, 1 month old. Oops, sorry. Oops, sorry. Why doesn't that work? Let me see, I hope it works now. Yes. So the father is just waving his hand in front of the eye of the baby. Baby doesn't blink. Baby only blinks when the skin is touched. So I do it again. So the father is waving, no eye blinks at all. And then he touches with his finger on the head, and Imke blinks. And now Imke is 3 months old. Father is waving the hand, and Imke is blinking, and she doesn't, no need to touch the face. So it's kind of, one has to learn that something which could be threatening to the eyes, that one blinks with the eye before the eye or the head is touched by the thing going towards the head. And that, yeah. Now, in this patient born without the cerebellum, she never had a cerebellum, you would assume that she would never had the opportunity to acquire this, if this is really a form of associative learning. And this is the case. So here it's my hand waving in front of her eyes, no blinks at all. No blinks at all, very different to Imke at the age of 3 months. Unfortunately, I didn't take a video touching the head of patients. When you touch it, then she gets the blink. So no cerebellum, no visual threat eye blink response. She had the disease from day 1 of her life. Now what happens in other patients who acquire the disease when they are 40 or 50? And we did a study of this. Here we test the visual threat eye blink paradigm with this paradigm. And what we do is, what happens is, so this ball is approaching the head, and the eyes are closed prior to the touch of the face. And we kind of can measure whether the eyes are closed prior to the touch of the face. This would be the conditioned response. And we did it in a group of controls and cerebellar patients. And we simply, we did it 20 times, and we just calculated the incidence, how often we saw this visual threat eye blink response. And in young controls, it's almost 100%. In elderly controls, it is a little less. Purkinje cells decrease around the age of 40. And then in patients with cerebellar degeneration, the CR incidence is much less, around 20%. So this kind of simple study shows that you could interpret it in a way that this conditioned response acquired early on, when you develop a cerebellar disease later on, that the memory goes away. So the cerebellum is involved in acquisition and in retention of this simple associative motor learning paradigm. I just want to give you one more example of eye blink conditioning, and this is in patient is the wrong word, but in children and young adults who have been born very preterm. And very preterm means that they are born earlier than 2 months prior the regular time of birth, or they have less, they have a weight of less than 1,000 grams. And we did eye blink conditioning in a group of children born very preterm, compared to children who have been born at term, preterm blue and at term green. And we did 1 session of eye blink conditioning, and neither of them conditioned very well. And then they came back for a second session, and then the term-born children, they learned the eye blink, and the preterm, they were much worse. So in children, it takes a little longer to acquire the conditioned eye blink response. So these were children born 5 to 6 years old. And then we looked also at young adults who have been between 18 and 25 at the time of the testing, and they have also been born very preterm. And this deficit, so this is these are the controls, and here are the patients. This deficit of reduced eye blink conditioning prevails, so it doesn't go away with during development. In very preterm, in prematurity, it is known that the cerebellar development is impaired. So during the last 3 months of development of the cerebellum prior birth, the granule cells, they increase in numbers at a large, very, very large extent. And they do it up to the time of birth. So a lot of developmental things in the cerebellum happen prior birth. And in these very preterm born children, this is very well described that there is problems with cerebellar development. In eye blink conditioning, it's very sensitive to cerebellar disease, and this is one example of it. Sam Wang, he put out the idea that when you have a lesion of the cerebellum very early on, that you get something which is called developmental diagesis. So when the cerebellum isn't working properly at the time of birth, that connected areas of the cerebral cortex don't develop very well. And motor areas, but also cognitive areas. And this might explain that, going back to this patient with cerebellar agenesis, she had motor problems, but these motor problems compared to other diseases which occur in adulthood are surprisingly small, but she has significant cognitive impairment, more than someone who gets a cerebellar disease in adulthood. So she had an IQ of 66. So this is kind of, yeah. Okay, but this is kind of a side. This is not the main topic of my talk. I just wanted to put this in because of these prematurely born children. So there is good evidence that the cerebellum is involved in motor learning. On the other hand, patients with cerebellar disease, they are treated with physical therapy, and this is about motor learning. So when you train your motor function with a physical therapist, you are treated with training of movements, and if you want to improve by it, you need to be able to do motor learning. And how come that patients with cerebellar disease are treated with physical therapy? They are treated with it, and right now there is no anti-ataxic drug. Genetic therapies are just beginning to be tested in clinical trials, and what really patients, the mainstay of treatment is physical therapy. For a long time, there haven't been any controlled studies to show that this helps, and it does help. So this is one of the very first studies looking at it. ZARA score is a clinical score of ataxia severity, and these are 4 time points: 8 weeks before training, just before training. Then the patients received 4 weeks of intensive physical therapy. This is after training, and this is 8 weeks after the end of the training. And the ZARA score, when it goes down, the clinical ataxia signs go down. So they improve. And this is group data showing it as well, before and after training. There's improvement of the ZARA score, and it stays. These are the patients with pure cerebellar disease. How come when the cerebellum is involved in motor learning? We did 2 studies for this, and this is one study where we did a balance task. So the patients with cerebellar disease received 2 weeks of balance training on this platform, where they had to move their center of gravity to different points or aims shown on the monitor. And they did it for 1 hour each day. And we did MRI scans prior training, after training, and then after 3 months. And patients got better. These are 2 measures. We had one called directional control, so how good they were able to aim to the targets. And in dark are patients, in gray are controls across 10 training days. And patients perform below controls. Yep, these are cerebellar patients, but both groups improved. Here are the number of falls. Controls hardly fall at all. And here in patients, and they reduced the falls. They wore a harness, so nothing happened, but the number of falls were reduced. So they learned, both groups learned. And then in MRI scans in the patients, we didn't see significant changes of gray matter volume in the cerebellum. So this is voxel-based morphometry analysis. However, what we saw is in the patient group, but not in the controls, in the patient groups, we saw a significant increase of gray matter volume in premotor cortex. So a connected area to the cerebellum in the motor control loop. This was about balance control. And then we did more recently a study of arm movements. So simple task where they had to point, to point a pointer to a start position and then to different targets. And they did it for 1 hour each day for a week. And before and after, we had again MRI scans. We had 4 different groups of training conditions, and I don't want to go into details because we didn't see any difference between the 4 training conditions. But this is why we have control group and cerebellar group with 4 conditions. And this is pre and post training. And this is an accuracy measure. And when you look at the controls in the 4 training groups, they are better, they perform better as the patients because they are not cerebellar-like, but they improve. So it's the error measure, it's going down. And this is a difference post-pre, and when it is negative, it's an improvement. There's more variability in the patient groups, and intercept is higher, but most of them improve as well. So we as before, for the balance task, we saw improvement. And then the voxel-based morphometry, we saw exactly the same thing as for the balance task. So in the patients, but not in the controls, we saw a significant increase of gray matter volume in the premotor cortex. So it seemed to be that other areas of the cerebellar thalamic cortical motor loop try to make good for the cerebellar deficit. So it seemed to be, at least based on the VBM analysis, that training effects to large extent are not due to improvement of the cerebellum itself, but of extracerebellar areas. That said, there are newer mouse studies out where they do motor training very early on. These patients have many years. There are many years in their disease of the cerebellar degeneration, but there are mouse studies where they did training very early on. And when you do it very early on in the disease and you have intense training in mice, then you can slow down or even improve the degeneration in the cerebellum itself. But here we are not early on, so that's a different story. This gray matter increase in premotor areas is seen not only related to the training task, it's there already at baseline. So what we did here is we compared the gray matter volume of the cerebellum. So this is gray matter volume projected on a flat map of the cerebellum, and the greener it gets, the less gray matter volume is there in patients. And this is expected. I mean, they have cerebellar degeneration, so we see atrophy. So there's nothing else in atrophy. So this is cerebellum smaller in patients than in controls. However, in the cerebellar cortex, we see that patients have more gray matter in the premotor cortex. So this seems to be something what cerebellar patients do all the time in their disease, so that this premotor cortex is trying to compensate for cerebellar deficits. Now I want to jump from motor learning to learning in the emotional field. And I started off with eye blink conditioning as a simple associative learning task in the motor domain. And now I go to classical fear conditioning, and we use simple fear conditioning task. And in this task, so when you do fear conditioning in humans, usually differential conditioning is done. So in eye blink conditioning, you have one CS and one US. It's just one tone, and you have your air path, nothing else. In fear conditioning, the community, usually in humans, not in animals, in humans, they present one CS called the CS plus, which is coupled with a shock as the US, and a CS minus, which is never coupled with the US. And then the difference between these two in behavior or in fMRI activity is compared in order to control for unspecific effects. This is not done in the animal literature for most of the time, and really the CS minus, you could argue this is learning a safety signal. So in a way, that's not neutral. But in any case, this is what people usually do, and this is what we did in this study. And this is in fMRI studies in healthy subjects, and we did it in the 7T scanner in Essen. And we measured fear responses. We measured it we used questionnaires, but we measured it also with skin conductance response, which is really, really standard. The thing with fear conditioning, different to eye blink conditioning is, and this is like people usually do it, is that usually in the fear conditioning field, a CS is used, which is presented for a very long time. So eye blink conditioning works best when you have very, very short CS-US intervals. So in humans, 500 milliseconds. 500 milliseconds, you can't measure bolt response. This takes 4, 5, 6 seconds to build up. But in the fear conditioning literature, they show the CS for 8 seconds, for example. This is standard interval. And there you have enough time to look for your bolt responses in an event-related design related to the CS. So you can look at bolt responses after presentation of the CS prior the US. So you can look at quote, quote, predictive fMRI activation. At the same time, you can look for bolt responses related to the US, the shock. And in fear conditioning, in order to make it a little bit more difficult so that participants don't learn it within 2 or 3 trials, usually the reinforcement rate is not 100%, but partial. So in eye blink conditioning, usually you have 100% reinforcement rate. That is a typical thing to get good eye blink conditioning. In fear conditioning, usually you have reinforcement rates of 70% something. So out of 10 trials, 3 trials of the CS Plus aren't paired with the US. And these trials, which are unexpected to the subjects, in these trials, you can look at the time where the US is expected and doesn't come. So you have something, you have a way to check what happens when the US is expected and doesn't come. So an unexpected omission of the US. Usually, I have to say, people in the fear conditioning field never looked at this time. Thomas Ernst, the doctoral student, the PhD student who did it, he looked at a very systematic way at it, not really knowing what he expected, to be honest, but he did it here, and it turned out this is our most interesting result. And this I will show next. First thing, fear conditioning works in the scanner, and I only want to show this here in more detail. So these are skin conductance responses here in the acquisition phase. White column CS plus, gray column CS minus, and you see there's a difference. There's less skin conductance response to the CS minus compared to the CS plus, and this difference is highest at the end of the acquisition phase. And here we have an extinction phase. At the beginning, there is a differential response, larger skin conductance response to the CS plus compared to the CS minus, and this is gone at the end of extinction phase. So it worked. We have fear conditioning in the scanner. Now, the fMRI results. Again, this is a flat map of the cerebellum. These flat maps haven't been developed by us, but by Jørn Dietrichsen, who is in London, Ontario now, and it's used by many, many people in the field. And this is easier to look at where you are in the cerebellum. So flat maps, when you look at your anatomy book of the cerebellum, flat maps are shown. So here, this is a flat map of the cerebellum of the vermis and the hemispheres. And then there are 10 lobules of the cerebellum. Here, this is the anterior lobe, lobules 1 to 5. And then we have the posterior lobe, lobules 6 to 10. And what is shown here is, this is the fMRI activation related to the time point presentation of the CS. CS plus larger CS minus, so it's always differential activations. And this is the time window where, when you do fear conditioning properly, where you see the CS and you expect the US to come. So you predict the US. And we see cerebellar activation, which is kind of expected. So the cerebellum is an associative learning machine. It's important for eye-blink conditioning, and in a way, it doesn't matter for the cerebellum what needs to be associated. And here it's a CS and an aversive stimulus. And here, this is the time, these is the average of the fMRI response of the unreinforced CS plus trials in the acquisition phase. So at time point where the participants expect the US to come, the electric shock, but the electric shock is not coming. And here we have much more activity in similar areas. And in addition, we see the vermal areas. People who do fear conditioning in animals, they record here an 8 because in vermal lobule 8, which is important for freezing. People, when you do fMRI studies of the cerebellum of emotional tasks, so you show angry faces or something, then most activation is here, intermediate part of the cerebellum. And then we have here the lateral areas, higher cognitive areas. So in fear conditioning, a lot of things happen at the same time. It's not only autonomic nervous system. And we see a huge spread between autonomic system, motor system, emotional system, and cognitive system in the cerebellum. But the main message here is cerebellar activation is highest in a situation where something is expected and doesn't occur. And you could argue that this reflects that it has something to do with processing of prediction error. And this activation goes away at this time point. US expected doesn't occur, it goes away in extinction. And extinction are several trials where the US doesn't occur. Now, you can take this interpretation one step further. There is the idea out that, so you could think about these single trials which are not reinforced in acquisition. You could think about them as initial extinction trials, because in extinction, yep, has come, but it's not reinforced anymore with the US. So you could think about it as initial, at the very first extinction trials. Extinction learning, there is an idea out that extinction learning has something to do with unexpected reward. So when the shock is not coming, this is kind of rewarding. It's nice, you don't get the electric shock. And these reward signals in the brain start off in the VTA, the ventral tegmental area. It's a dopaminergic signal. And there are animal studies, recording studies of the VTA done by Kalisch, but also by others. And what they found is, in the very initial extinction trials, where the US is expected and doesn't come, when they measure at the time point of the US omission, the unexpected US omission, they have very high VTA signal. And this fades away during extinction. What we are studying in the lab right now is there are findings, there are old findings that the cerebellum receives afferent input from the ventral tegmental area. This is old. New findings is that the cerebellum has a direct connection, the cerebellar nuclei, to the VTA. And in addition, there are more and more studies out which show that the cerebellum does not only receive sensory information, but also information about reward. So there are studies out showing climbing fiber signals that there are climbing fiber signals which are related to rewards, unexpected reward omissions, reward predictions. So there are animal studies show, And what we are interested right now is, but I can't show you data. This is really, we are running 70 experiments right now to support the hypothesis that the cerebellum contributes to prediction error. That's not new. That it contributes to reward prediction error. This would be new. And prediction errors are necessary for learning, and that in the extinction learning of in fear conditioning, that the cerebellum is involved in reward prediction error processing necessary for this extinction. And my last slide here is that this what we saw in fear conditioning, that this might be one further example showing that this very famous scheme put forward by Doria, that there are 3 different systems contributing to motor learning, the cerebral cortex, important in use-dependent or unsupervised learning, the cerebellum important in supervised learning, and basal ganglia and reinforcement learning, which is reward-related, that this is not wrong, but too simple, so that the cerebellum is also doing something for reinforcement learning. And with this, I would like to thank you for your attention.
Amazing. Thank you very much for this beautiful talk. Let me open it up to the floor and see if there's questions in the audience. I see a lot of clapping. I do apologize if I mistake a clapping for a raised hand. But oh, yes, there is a question. Valentina.
Hi, thank you so much for your talk. It was very interesting. And having a background in neuropsychology, it was very, very interesting. So I have a question on the relationship between the cerebellum and the premotor cortex, and in particular, how the activity between the 2 structures is organized in a normal brain, and like how they combine the activities to weight in performance learning or motor performance in the healthy brain. And is there any hierarchical interaction between the structures?
So the connection between the cerebellum and premotor cortex is very well known via the thalamus. So it always goes via the thalamus. So this is very well known. And it is known that the cerebellum contributes to motor planning via the premotor cortex. So it's kind of a very well-known loop, in addition to the loop to the primary motor cortex.
Okay, thank you.
So whilst other people are trying to formulate their questions, I'll chip in with my list of questions. And the first question I have for you is for your eLife study in 2019, the fear conditioning. You mentioned that you used 7T imaging, and I was wondering what the rationale for 7T versus 3T or even more clear.
Yes. Yeah, I can give you the rationale. Also, it didn't work out for us. So our idea was in the animal literature, it's very well known that the fastigial nuclei are important for fear conditioning. So this is when you do rodent studies of fear conditioning, it's either lobule 8 of the vermal cortex or the fastigial nuclei. And this is known for a very long time. So our hope was that we could see activation of the cerebellar cortex and the nuclei. However, our signal was not strong enough. So for 3T, 70 has a higher signal to noise ratio. And we did fMRI studies with motor task, finger movements or something, in the 70 and idling conditioning, and we were successful. So we were able to activate dentate nucleus, but it didn't work out for the fastigial nuclei. Yeah, that was the rationale. And now the 70 scanner, we have a new one in Essen, and with stronger gradients. And we continue with fear conditioning, answering these questions for extinction. And we hope that we get the nuclei. Plus, in addition, a very interesting thing, and I didn't bring these things together for a long time. So we did fear conditioning in patients with cerebellar degeneration. So who had atrophy of the cerebellar cortex. And you would think, yeah, you see less activation in the cerebellar cortex, which how much would that help you when you have cerebellar degeneration? But this is not what we saw. We saw the activation level wasn't different between patients and controls. And I think the reason for this is that what you see in the cerebellar cortex is a synaptic input. So fMRI activity reflects synaptic input, and the cerebellum gets a lot of synaptic input to the granule cells. And in the patients we tested, they are models of Purkinje cell degeneration and output. And I think the input is coming in, and the input is good. It will not use, in patients with cerebellar degeneration, they will not use the input properly. So I think what we see is incoming information. For example, incoming reward prediction error, like people who record at the granule cells or in the climbing fibers, they see this, but this is incoming information. It doesn't mean that it doesn't show you what the cerebellum does with it. So I think what we see in the cerebellar cortex is really input coming in. And our hope is now with this improved 70 scanner, and we do a larger N. So we now go to 50 subjects to increase it even further so that we have the cerebellar cortex input, and then the cerebellar nuclei, which is the output, receiving the synaptic input from the Purkinje cells, so indirectly. And this is, I think, this is why we do it at 70.
That sounds very reasonable to go for 70 in that case. The other question I had was a point of clarification, and I may have misunderstood that in the beginning, but you mentioned that the patients with cerebellar agenesis are very rare, and you mentioned a number 10 to 20. And that
at least described in the literature. So with known patients described in the literature, maybe there are 100 worldwide, but yeah.
So how does cerebellar agenesis come about? Do we know, or is it a coincidental finding? And you mentioned that they're now more detected with imaging, but surely there are behavioral differences that would identify them.
Yeah, this is true. So these, answering the last question first, so these children have a delayed motor development, even though, the patient I know, she was 2 or 3 years old until she learned to walk. And there are descriptions of children who were 9 years old until they learned to walk. And nowadays, when you don't walk by the age of 2, you get diagnostics. Mm-hmm. And then very early on, you get an MRI scan, and then you see it on the MRI scan. My patient, in fact, she grew up in post-war Germany in a very rural area. She came to the doctors at the age of 50 because she had vertigo and tinnitus, and the ENT people did an MRI scan, and this was just by chance. But now it wouldn't happen. I mean, now when you don't walk properly by 2 or 3, you get an MRI scan, and this is why more cases are described in the literature. And it's not that clear why. So some people think that is that this is hereditary, but the gene mutation hasn't been found. And the other idea is that something happened, like an infection or something, very, very early on, and that the cerebellum was unable to be developed. But this hasn't been solved yet.
Right. And so coming from my own background, I looked at patients with agenesis of the corpus callosum, so complete or partial absence of the corpus callosum. And obviously having a white matter background, I was wondering in patients where you have agenesis of the cerebellum, does it equally impact the white and the gray matter? Is there a difference?
You mean of the cerebral cortex, of the connected areas?
Yeah.
I don't think that this has been done properly. You would think so. I mean, one would think so. I mean, we didn't do it. We didn't do it. But yeah, I think so. Like in someone, yeah.
Right. There's a study to be done.
Yeah.
I can see that Ahmed raised his hand.
Yes, hi. This is all very interesting, and I think as many of us are not so familiar with the cerebellar literature. But so my question to you is about one of the slides you showed with premature infants, and you showed that their learned response is quite delayed, even at 5 or 6 years old. Is that what you showed as a follow-up time too?
Yeah, but in both, in children born at term and in children born preterm, so they need longer. So this is just children need longer time to develop the conditioned eye blink response. So when you are a young adult, then when you are a good conditioner, you need 20-30 pair trials, and it's there. But when you are 5 to 6 years old, even though when you're completely healthy, you need more than 100 trials. So on the first day, when we had 100 pair trials like we do it in adults, we didn't see a difference. And even the healthy ones, they didn't develop eye blink conditioned eye blinks. And then we called them in for a second time. And then the term-born children, they showed conditioned responses and the others not. So there is, at 5 to 6, you're not as good in eye blink conditioning as compared to when you are 20.
And so do we know if this ever catches up in those preterm infants? And do we know if there are other things? Okay, so even later on in life. Okay.
Yeah, so we didn't follow up the same preterms. So we had a group 5, 6 years old, and then we had a group 18 to 25. And the 18 to 25, so 18 to 25 term born, within 20-30 trials, they have conditioned responses. And the preterms, they were slower in learning and they never reached the levels. So it's not gone. It stays. Yeah,
right. Okay.
Stays.
Okay. So then, I mean, this is of course one particular test, but I imagine that, this has other effects that haven't been discussed.
Yeah.
At the cerebellar level or otherwise. Yeah. Okay.
Yeah.
Okay.
Yes, it has. So in preterms, I mean, 20 years ago, these very preterm children, they died because of lung problems. Now, the pediatricians are so good that they survive. And now they see that one thing is, for example, the blood vessels in the cerebellum. When you are preterm born, they're not as good and they can get bleedings and stuff. But even in and then you get cerebellar lesions. But even when this doesn't happen, you don't have bleedings, nothing, the development of the cerebellum is not as good. So the volume, I mean, when you have an MRI scan, a regular one, you don't see nothing. But when you measure the volume, there is less volume. So the cerebellum is one hotspot of problems of these very preterm born children, but not only. I mean, when you look at white matter, there is a lot of problems with white matter. So other areas have problems as well. But because the cerebellum, it's a very sensitive area, time before birth for the cerebellum, it's a hotspot. And this, I mean, very preterm children, even though when they are very well off, they tend to be clumsy, and they tend to be that they have problems in school. So there are always high performers. I know one professor in cerebellum field, he is very preterm, and I don't see any problems, but on average, you can see it, there are problems. And people in the field, they think that the cerebellum contributes to it. And there, the earlier, I mean, when you get your cerebellar lesion around the time of birth, the cognitive and emotional deficits are much more prominent than the motor deficits. And when you get your cerebellar disease, as a rule of thumb, as an adult, it's the other way around. So when you are an adult neurologist like me, motor problems is the first thing, why they come, and the cognitive problems are much less. And this is not good understood why this is. Yeah.
Right. Okay. Very interesting. Thank you.
Actually, quite interesting if I can just intersect here, because I work a lot with stroke patients, and obviously when you damage an established network, there's different mechanisms of how you might be able to recover some of the function. But when we look at the children that suffer a stroke to the brain, then the brain is quite good at adjusting to it and not causing a permanent deficit. So it seems like the cerebellum is less flexible to compensate in that way. Is that fair to say?
Yeah, I think that is a very good question regarding the cerebellum. I mean, one thing is, it seemed to be really you need a proper cerebellum to develop your frontal cortex properly for cognitive and emotional function. So when your cerebellum is malfunctioning or not there during the time of development of the frontal cortex doesn't develop that good. And there are animal models now showing this as well. On the other hand, you could argue that they show less. I mean, this patient without the cerebellum, she's able to walk without walking aids. And if you would take out the cerebellum when you are 40, you would be unable to walk at all. You would be wheelchair bound. So it may be that motor function that they can compensate, but this hasn't been studied properly. So it might be a thing of location in the cerebellum. It may be that newer areas of the cerebellum that they need longer to develop, and they still develop prior birth, and motor areas are developed earlier, so they are less affected by preterm birth. That might be one thing. And the other thing, it might be, so maybe some things they can compensate, and there might be critical time windows of development. So I guess these strokes, when they happen when you are 5 or 6, that might be a completely different story as compared to it happens around birth. And it might be around birth. Initial developmental steps are very important, and they don't get it. In 5, 6, your brain is very plastic, and you can something like this.
It's those critical time windows, isn't it?
Yeah, it might be. Yeah, I would. But it hasn't been studied in a systematic way. So these are just you need people now to study it in a very systematic way.
Seems like we're coming up with topics for the next 5 to 10 years here. Lilith has a question for you as well.
Yeah, hello, thank you. Can you hear me? Okay, thank you again for the talk. It was really interesting. I have 2 questions. So the first one is, for example, I don't know, we have a patient with a degeneration or some type of lesion in premotor cortex. Does it mean that training will, the training of cerebellum will increase the gray matter volume in that area?
Good question. You would think so, that this is a very good assumption. I don't know if anybody has ever done it, but yeah, I think that would be a good guess that when your premotor cortex is lesioned, that the cerebellum tries to help out.
Thank you. And then the second question, did you do the follow-up study of those people who increased the the gray matter volume in premotor cortex. So I was wondering how long-term is that effect, or it just
Yeah, in our first study we did it after 2 or 3 months, and it was still there.
Okay.
And in the second study we didn't have a follow-up.
Okay. Thank you very much.
I wanted to pick on a study you presented as well, the HPM 2020 study, where you showed that there was more variability in the behavioral measures in the patients compared to the controls. And you also did the imaging, so I was wondering if you also found more variability in the imaging measurements.
We haven't looked at it, but this is a very good question. You would think so, but we haven't done it. Yeah.
Here you go, another one to add to the list.
Yeah.
Fantastic. The last question, well, 2 more questions if I may. First to last, are there any laterality effects that you became aware of?
For training or motor?
Because we think of the brain as very lateralized depending on the cognitive function that we look at, and then especially imaging tends to ignore the cerebellum most of the time because it's often not even in the templates. So I was just wondering what your experience of laterality was.
So let me remind myself, because it was the premotor cortex in both studies with this balance training and with the arm movements. The finding was strongest on the right, very little on the left. I mean, they used the right hand. You would expect opposite side, which would be left. And for the balance task, this shouldn't be lateralized. So there is some indication that for some reason the right premotor cortex shows more changes than the left, but I don't have a good explanation for it. And I'm always a little bit cautious with these things. And I mean, for cerebellar research, we have a decent number, but it's a small number. And I think it would need to be replicated in a larger group.
Fair enough. And then really my final question for you today. So I come from language sciences, broadly speaking, and there has been a couple of publications over the last years saying that the cerebellum is also involved in higher cognitive functions, including language. And I was just interested in hearing your opinion on it.
Yeah, I think that's correct. So I'm kind of I have a very conservative and slowly functioning mind. So for a long time, I thought this is not right. Cerebellum is important for motor control. And when I see my patients, I don't have any cognitive problems, but that's not true. Yeah, so I think Jeremy Schmarman, he got it right, I have to say, because although patients in an adult clinic, they come with motor dysfunction, but I see patients over many years, and when they have a degeneration, almost all of them develop cognitive signs, and very many, you have to ask them, and very many say that they have problems with, that they often have to search for words, and it takes them a long time. And so I think it's right, the cerebellum contributes language function, and this is lateralized. So there are so many fMRI studies out now, and this is lateralized to the right cerebellar hemisphere. So I think, yes, this is correct. And in a way, the cerebellum is a mirror image of the rest of the brain. So any kind of brain function is supported by the cerebellum. Yeah.
That's a quite a nice snippet as a concluding remark, I guess. Thank you so much again for being here today and presenting your amazing work. It's absolutely fascinating. Thanks everyone who was live here on Zoom with us and live over on YouTube. just to let you know, there won't be a talk next month, but we will be back in November with finding the network balance in Parkinson's hallucination. So do join us in November, as always, the last Thursday of the month. And until then, have an exciting time in science, and we hope to see you soon. Bye.
Thanks a lot for the invitation. Bye-bye. Hope to see you soon.